Welcome to our lesson on mitosis, the essential process of cell division that fuels growth, repair, and regeneration in every living organism. Mitosis is a cell's way of making copies of itself. It occurs when a cell needs to grow, repair damage, or replace old cells. Before dividing, the cell prepares by gathering all the necessary materials to split into two new, identical cells. There are several key stages in mitosis: interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Together, these steps ensure that each new cell is an exact genetic copy of its parent. Before mitosis begins, the cell enters interphase, a crucial preparatory phase. During interphase, the cell grows, duplicates its DNA, and readies itself for division. Although it isn't technically part of mitosis, interphase lays the foundation for the entire process. Mitosis officially kicks off with prophase. Here, the cell's chromatin, a mix of DNA and proteins, condenses into visible chromosomes. Each chromosome forms two identical sister chromatids joined at a region called the centromere. The nuclear envelope starts to break down, and spindle fibers begin to form, setting the stage for division. During metaphase, chromosomes align along an imaginary line known as the metaphase plate, which is equidistant from the cell's two poles. This perfect alignment ensures that each daughter cell will receive an identical set of chromosomes. In anaphase, the sister chromatids separate as the spindle fibers attached to the centromere shorten. This separation pulls the chromatids toward opposite poles of the cell, guaranteeing that each new cell will have the same genetic material. Telophase is marked by the arrival of the separated chromosomes at the cell poles. New nuclear envelopes form around each set of chromosomes, which then begin to decondense back into chromatin. Meanwhile, the spindle fibers disassemble, signaling the near end of cell division. The final stage, cytokinesis, divides the cell's cytoplasm into two separate daughter cells. In animal cells, this is accomplished by a cleavage furrow that pinches the cell membrane inward until the cell splits completely. After cytokinesis, the two daughter cells enter interphase and the cycle may begin again. Each daughter cell is genetically identical to the parent cell, containing the same number of chromosomes. Mitosis is vital for growth, development, tissue repair, and asexual reproduction in multicellular organisms. Thank you for joining us on this journey through the stages of mitosis. If you enjoyed this video, please like, subscribe, and share for more exciting insights into the incredible processes of life.